Afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Camille Hoffman will present Choline, a new prenatal supplement to improve a child's mental health. The Brain and Behavior Research Foundation funds the most innovative ideas in neuroscience and psychiatry to better understand the causes and develop new ways to treat brain and behavior disorders. These disorders include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, PTSD, and schizophrenia. Since 1987, the Foundation has awarded more than $408 million to fund more than 5,900 grants to more than 4,800 scientists around the world. 100% of all donor contributions for research are invested in our grants to scientists. I'm delighted to introduce Dr. Camille Hoffman. Dr. Hoffman is Associate Professor in the Departments of Obstetrics and Gynecology and Psychiatry at the University of Colorado School of Medicine. She was the Foundation's 2015 Baer Prize winner for innovative and promising schizophrenia research. Today's webinar will begin with Dr. Hoffman's presentation. This will be followed by a question and answer period. To submit your questions, please use the Questions tab on the control panel on your screen. Feel free to submit your questions at any time. Following the presentation, I will ask as many as possible in the time allotted. And now, I'm pleased to introduce Dr. Hoffman. Camille, the floor is yours.
Thank you so much, Dr. Borenstein. And I am grateful to the Foundation for receiving the Baer Prize that you mentioned back in 2015, and also very excited to present my research today on choline.
If you can just click on this slide to advance it, please.
There we go. So as you mentioned, I've received some financial support from the Brain and Behavior Research Foundation, as well as some local foundations in Colorado, as well as National Institutes of Health. And I nor any of the researchers in the group that I'm going to mention today have any financial disclosures. And I have to acknowledge at the start this man, Randy Ross, a psychiatrist who was well known to the BBRF as well as to the world of choline research, who passed away in December of 2016. He was really a pioneer of this research, and so it's been my my privilege to continue the work that he started. So I was going to briefly talk about today the developmental origins of health and disease hypothesis, or DOHADS. We think about that a lot in obstetrics and in high-risk obstetrics, but as it pertains to mental health today, assess genetic and environmental factors that impact fetal brain development, those that are known at present, and then describe how choline improves human brain development starting prenatally. And this started really almost 100 years ago, or 100 years ago, based on this National Child Welfare Association poster, which acknowledged that the perinatal experience influences maternal outcomes as well as pregnancy outcomes and how that translates into her child's development. We know in the short term that mood disorders, stress, substance use impact pregnancy outcomes like preterm birth and low birth weight infants, both of which are markers for adverse mental and physical health over the life course, as well as predisposed to perinatal mood disorders. And then that later in the life of the child, these adverse pregnancy outcomes, as well as other prenatal experiences, have been related to mood and anxiety disorders, cognition disorders, behavioral and developmental disorders, autism spectrum, schizophrenia, other chronic medical diseases of adulthood, and shortened telomere length. And that can play out over the life course of the child. And in fact, Leonardo da Vinci even had this notion back in the 1400s when he voiced that the things desired by the mother are often found impressed on the child that the mother carries. So I think even he had the notion of the DOHAD hypothesis as it pertains to mental health. So we know in obstetrics and over life that we are all kind of a combination or culmination of nature, which is our DNA, our genetics, and nurture, which can influence our expression of our genetics, as well as other ways that we manifest illness and wellness. And this group out of Switzerland nicely summarized the way that brain function, our central nervous system function, can be influenced across the life course by the nature and nurture components prenatally. So we start with our maternal and paternal genes that we inherit at the moment of conception. But then we also know that there are multiple prenatal influences. So MIA stands for inflammation and other stressors. But other factors like alcohol exposure, marijuana exposure, infection exposure can also be big influencers of fetal development, including fetal brain development, where early insults that are severe can sometimes result in severe brain or other central nervous system malformations and resulting in miscarriage or premature death or stillbirth. Going down a little bit in pregnancy towards delivery, perinatal or early postnatal factors or insults can result in cerebral palsy. Earlier in childhood, prenatal factors may start to manifest along the autism spectrum. We know that other conditions like schizophrenia, depression, bipolar disorder, ADHD can manifest earlier in childhood, but often in puberty and post-puberty or early adulthood. And even towards the latter part of the life course, that age-related disorders like Alzheimer's or Parkinson's disease or other causes of dementia may be part of the end of that spectrum. But that even these may have origins that started prenatally and then were shaped and sculpted into various conditions or avoidance of conditions over the course of that individual's life. So prenatal factors that I've kind of alluded to, but that we know have the greatest impact on a child's or fetus's brain development are maternal illnesses, particularly infections that instigate inflammation and cytokine activation during pregnancy and in the second trimester of pregnancy, appearing to be the most vulnerable time. Then maternal alcohol, tobacco, and marijuana use, maternal nutritional deficits, maternal psychosocial stress and stressors, psychiatric symptoms or conditions, diagnoses, and then genetics that can play throughout all of these exposures, and obstetric complications, and that is primarily focused on the time of labor and childbirth. And we know that at least as it pertains to schizophrenia, the combination of nature and nurture and that body of science has been well developed. So we know that from the genetic aspect, there are many genes that are associated with schizophrenia, and these are expressed at the highest levels in the fetus and his or her placenta, the organ that connects the fetus to the mom's uterus. The familial odds ratio, if an individual is affected by one of these genes, is about fourfold, and that contributes to about 10% of the cases of schizophrenia. In our group with Dr. Ross and people who have also since retired, started looking at this particular gene, CHRNA7. Its deletion has an odds of 15-fold in development of schizophrenia, although it only contributes to about 0.05% of the cases of schizophrenia. But I mention this because this gene and the way that it functioned formed the basis of a lot of the research that we now do in relation to choline and how it interacts with the receptor that this CHRNA7 gene produces normally. And then nurture factors, so maternal environment. I mentioned that infections, in particular respiratory infections, and sexually transmitted infections are pretty large contributors to later development of schizophrenia and psychosis in the child. Cigarette smoking is a contributor, although that may be biased by the likelihood of people affected by schizophrenia to use tobacco. And then prenatal marijuana use is the more recent data that our group is focusing on, and a lot of other groups. We know from our data and others that it has some association with a decrease in childhood cognition, and maybe some markers that could translate into a vulnerability towards psychosis later in life. And then malnutrition, but these kinds of malnutrition states have to be pretty severe. So the data that we have in relation to states of malnutrition being related to schizophrenia come from Holland and the famine in World War II and the China Cultural Revolution. So not a small amount of weight loss during pregnancy, but real states of malnutrition with famine. And scientists have really understood for at least 20 years that early brain dysfunction is identifiable in newborns, even though when identified, luckily the vast majority don't develop schizophrenia. Individuals with schizophrenia can be kind of traced back and some early markers be noted retrospectively by their family or by their family doctors to have manifest as early as infancy. So I want to back up a little and talk about TRNA7 and give you a little bit more background on that. I tried to make it as concise as possible, but this is failing to acknowledge many, many people who have put forth decades of research into getting this slide, the data that I'm presenting for you on this slide. So it's a gene that makes the alpha-7 nicotinic acetylcholine receptor, one of our major neurotransmitters in our brain and central nervous system. It lives on chromosome 15q13. That's the short arm of chromosome 15. And deletions in this gene, so subtractions of genetic material within this gene, have been linked to schizophrenia and autism spectrum disorder, where duplications, so extra parts of this gene added, have been related to ADHD. TRNA7 and the alpha-7 nicotinic receptor that it makes are involved ultimately in the fetal brain in inhibitory neuron function or dysfunction, and this relates to sensory gating abnormalities in psychosis. And this is a heritable defect in families with schizophrenia, although I already mentioned to you that it's overall a small contribution to the overall cases of individuals affected with schizophrenia. So in this figure, and I think I have it again here, this is an example of a fetal pyramidal cell, and the little cylinders lined up side by side represent the alpha-7 nicotinic receptor, and this is mainly to illustrate how rich a fetal neuron, in this case a cerebral inhibition neuron in the hippocampus on a pyramidal cell there, are when compared to an adult pyramidal cell. And so the ways that this works and transitions from fetal brain function to adult brain function kind of backs up to, in early gestation, GABA is an excitatory neurotransmitter. Although we always think about GABA as kind of a soothing or inhibitory neurotransmitter, in the fetus it's excitatory early in pregnancy. Fetuses have many more alpha-7 nicotinic receptors than we do after birth. They're necessary to help stimulate critical depolarizing potentials that help promote further brain development and correct architecture as the brain develops kind of layer by layer. These receptors are more widely distributed throughout the fetal brain and within the neurons. And there is little prenatal acetylcholinergic innervation to compete with choline as an agonist. And therefore, it is the primary agonist in early development for these alpha-7 nicotinic receptors, because people often ask, well, what about acetylcholine? Once acetylcholine occurs in any considerable amount, it outcompetes choline right away. And this is kind of what happens towards the very end of gestation and after birth. and why choline is so fetal brain specific. So in other words, and this is a slide that I got from Randy Ross, our prenatal brain develops using a series of neurotransmitter operating systems. And early in gestation, we install operating system 2 or OS2, and this is primarily driven by choline and the TRNA7 gene and its expression. Then by the third trimester, or when we're installing OS5, TRNA7 appears again in large quantity, and at this point, it leads to GABA becoming normally an inhibitory functioning neurotransmitter versus the excitatory state that it existed in previously. And the thing is that once installed, the final system is permanent and it can't be uninstalled. And so it's that transition via choline induction and stimulation of normal TRNA7 and alpha-7 nicotinic receptor activity where we are able to respond to GABA in an inhibitory kind of way. And inhibitory neurocircuits, just to give you a broad view for those of you not aware, are involved in working memory in our prefrontal cortex, in somatosensory specificity in our somatosensory cortex, in associative fear learning in our auditory cortex, and in recognition memory in the hippocampus. And this is just to name a few of its functions. And it has also been identified in schizophrenia, in bipolar disorder, and autism spectrum disorders, depression, PTSD, ADHD, Down syndrome, and Parkinson's. So pretty vast list of conditions that failure or incomplete transition to inhibitory neurocircuits can be involved in. So this kind of sets up the rationale for our initial studies that were, again, pioneered by Randy Ross, where we had these models of, okay, a fetus has rich alpha-7 nicotinic receptor expression. Choline is the agonist and helps promote good neurodevelopment as a fetus. We had identified that there were genetic and potentially nutritional deficiencies that could impede this process. And so then the scientific question became, can choline supplements overcome these deficiencies, one or the other? And back at this stage, our group was putting a lot of thought into, should we screen everyone for these genetic conditions and then target them? Or is it more reasonable and rational to look at choline supplementation more broadly, since by the time prenatally, we may have been able to do those type of genetic assessments on the parents, and it would be even harder to do those on a fetus because those would involve invasive studies. We may have missed the opportunity for choline to benefit if we were going to see a benefit. So just wanted to also give you a sense before I launch into data on what are causes of choline deficiency in pregnancy. We know that a small group of humans, of women, have a polymorphism in the phenylethylamine transferase, or PEMT gene, and so that has been associated with choline deficiency, or an inability to manufacture or process choline. And this is associated with schizophrenia via another gene, that there are some dietary deficiencies or dietary deficiency states, and tremendous amounts of choline are needed for cell membrane synthesis, to synthesize DNA that can go into the cell to make it function, and that choline is also the major agonist prior to acetylcholine coming onto the scene. And then we know that maternal stress and stressors has led to liver sequestration, especially in the first trimester in some cases. And then kind of looking at North American data over time, in any given study, somewhere between 20 to 50% of women fail to reach optimal choline intake during their pregnancies. And cigarette smoking is one thing that can also influence that. And an estimated 20% of women smoke cigarettes at some point in pregnancy, so another factor. And these were all things that we looked at when designing the initial pilot choline study. as things that could become barriers to our research and our result findings. And so we took them into consideration when we decided to just broadly recommend choline and randomize women to choline versus a placebo for our initial pilot study. And so this kind of goes back to other things that can influence choline digestion in the maternal gut and release into the bloodstream. Again, a genetic defect can also prevent good choline synthesis. Storage or taking choline out of the maternal bloodstream and sequestering in the liver can influence its availability. It has to be transported across the placenta, and the more data that we have on placental transport and the way that it responds to different maternal states, such as stress, the more we realize that we have a lot more to understand about how the placenta may have its own behavior. And then we have the genetics, the nicotine exposure, and what actually makes it across the placenta into the fetus and how the fetus is able to process and utilize it. So multiple different steps that we know are involved from taking a supplement to getting it all the way across to the fetus. These are some data from a different lab, which I think highlight that comment that I made about placental behavior a little bit more. So this group out of New York looked at 2 different levels of choline supplementation in the 3rd trimester. They gave women 480 milligrams daily versus 930 milligrams daily. And this group actually had those women's food prescribed daily, and I believe that they even went in and ate 1 meal a day in the 3rd trimester so that they knew exactly how much choline they were getting. And when they looked at comparing umbilical cord cortisol at the time of birth to maternal plasma cortisol, they found that higher maternal choline intake decreased umbilical cord cortisol levels, so decreased cortisol exposure to the fetus, now newborn, but there was no difference in maternal cortisol levels. And at the same time, they looked at a couple of different genes, but this one that I put on this slide, placental corticotropin releasing hormone, or PCRH, between the 2 groups. And on the left, or in the gray bar, we have the group that received the 480 milligrams of choline versus the black bar, 930 milligrams of choline. And placental CRH gene methylation, as well as gene transcription. And they found that gene methylation was increased with the higher choline intake group, and gene transcription was decreased in the higher choline group. And this means, again, that this was another way that the fetus was being buffered from receiving too much cortisol exposure, because more CRH methylation would downregulate cortisol production at the level of the placenta, and transcription would subsequently also downregulate production of that CRH hormone. So some evidence that while it may not directly affect the maternal plasma cortisol, choline does have a buffering effect in relation to choline level on the fetus and exposure to stress hormones. In other human trials of prenatal choline, the data are varied. One study found no difference in 5-month cognition related to maternal choline levels, although the average estimated daily intake in that group was less than 400 milligrams a day. And the kind of quotation control group on the previous slide was at 450 milligrams. Several observational studies have associated higher choline levels or a higher choline intake with improved child cognition through the first 7 years of life, so a little bit of variation there. And no study, more importantly, has found any adverse effects. The maximum tolerated dose of choline is about 7 grams, and in the type of choline that we use in our studies, which is the phosphatidylcholine type, that would be equivalent to about 49 grams of phosphatidylcholine, so almost 2 ounces of phosphatidylcholine daily, which would be quite excessive and well beyond the dosing that we use in our studies. So I wanted to give you a sense of the randomized controlled trial that we're currently doing. We've done the pilot randomized controlled trial, and the data that I'm going to present to you from here on out come from that and an observational study that we did. And our first pilot randomized controlled trial included 100 women, and our observational study included 250 women. And now we are about midway through recruitment for the second or subsequent randomized trial, where we're randomizing women by 16 weeks gestation, so just into the second trimester, to receive either phosphatidylcholine at a dose of 9,000 milligrams daily, which is equivalent to about 900 milligrams of choline daily, versus a placebo. This is a blinded study, so none of the research personnel or study participants know which branch they're randomized to. And then this is just an example of our study timeline, where we follow women and their fetuses with ultrasound, with hair measures, with blood measures, with surveys throughout the course of pregnancy, and then also at multiple time points in the postpartum period, out to 40 months of life for that child. And the primary outcome of this study is the Child Behavioral Checklist score at 40 months of life. But as you can see, there are multiple measures and multiple data points that we are collecting between 16 weeks and essentially 4 years, almost 4 years of life. So I mentioned this already. We're going for about 900 milligrams of choline in the form of phosphatidylcholine, and about 10% of phosphatidylcholine is bioavailable as choline. So that's where the 9,000 milligrams roughly comes from. And this is equivalent to about 3 to 4 extra large eggs per day. Our placebo capsules are identical. And for study and research purposes, we know based on some other studies that women receive dietary advice on choline-rich foods in both groups, and that is one of the reasons why we give them so much choline supplementation in the phosphatidylcholine randomized group, so that we can still see a difference even if women are meeting the dietary instructions that we give them. We felt like it was unethical to randomize people to choline or placebo, and knowing what we know about choline as a buffer, to not give them at least some guidance on how to achieve that in their diets. And then we do have a website that probably will answer a lot of your questions related to this study. It's prenataldoctoradvice.com, and any of you can access that for more information on our study and our research team. So now I wanted to show you some of the pilot data and then kind of the directions that we're going in based on the data that we now have. So this is from the first pilot randomized controlled trial, where we use slightly lower dosing of the phosphatidylcholine versus a placebo that was also identical. And the findings in really brief summary from that study were that infants who were treated with choline at 1 month of age had better performance on an auditory EEG called a P50, where they hear 2 sounds 50 milliseconds apart. And so this is to give you an idea of what I'm talking about and what we expect to see on a normal versus an abnormal P50 EEG. So the top is an infant who was happened to be treated with choline, and this is at 1 month of life or 1 month after birth. And to the first sound, which are little clicks that we get or that we play while the infants are asleep, the first response is notable with a peak and a trough. But then 50 milliseconds later, the same click is played and the response is suppressed. So this kind of goes to our ability to inhibit response to second sounds, or more globally, our ability to tune out background noise or background distractions in general. This is an infant who had an abnormal P50 study, and so here there's the peak and the trough to sound 1, and a pretty comparable peak and a trough to sound 2. And then in the bottom corner, I've kind of crammed in here an example of a P50 study in an adult who doesn't have schizophrenia versus one who does. And the response is similar. The control subject is able to pretty much entirely forget about or suppress the second sound exposure, versus the patient with schizophrenia has a nearly identical response. And we know that in conditions like schizophrenia or psychosis, it's the inability to turn off the background noise that is a constant distraction that is part of those illnesses. So to change those little wave patterns to bars from this pilot randomized controlled trial, we found that in low-risk mothers, and in this pilot, we were more focused on women who had pre-existing or known mental illnesses, women who had genetic predisposition, and then broke them up into low and high-risk mothers. But you can see in either group, the low-risk mothers or the high-risk mothers, and the blue are those who received the placebo, and the red are those who received choline, that there were a higher percentage of those with normal P50 ratios in both groups with choline exposure. So we determined that prenatal choline, at least at about 1 month of life, did appear to enhance P50 inhibition in newborns in both low and high-risk mothers. And then when we looked at those same children, or really a subgroup of them, because we lost some of them to follow up over the course of about 40 months postpartum, that the newborn P50 studies, so the ones above, were also predictive of 40-month-old behavioral scores on the child behavior checklist. So here in blue are P50 scores that were slightly elevated, and in red are the scores that are less than 0.5, so normal P50 ratio. And again, you can see that those who had normal P50s were less likely to score high on the withdrawn scale, attention, or total problems on the child behavioral checklist. So we all know that 40-month behavioral assessments don't directly correlate with or don't 100% predict disorders later in life. However, they have been modestly to better correlated with later issues, not always schizophrenia, but have also been correlated with ADHD, bipolar disorder. And then when we looked in some of our observational data, after wrapping up the first phase, or that pilot randomized controlled trial, at other factors that we knew were related to adverse neurodevelopment of the fetus, we looked at women who had reported any evidence of infection during the second trimester, and we asked about urinary tract infection, respiratory infection, sexually transmitted infection, and then corroborated that with the medical record, and looked at maternal serum choline at 16-week gestation. We had a similar finding on the P50, that there was enhanced P50 inhibition and regulation in in infants of mothers who had higher choline levels as early as 16 weeks gestation. And so the top figure is the P50 inhibition again in women who had infection in red, and you can see that their P50 inhibition, and here lower is worse, had poorer P50 inhibition in the lower levels of choline in serum at 16 weeks, versus as they increased their serum choline in micromoles, the baby's P50 inhibition improved, and this was assessed at 1 month of age. And then at 1 year of age, we used an infant behavioral questionnaire, IBQ, and also saw improved IBQ regulation at 1 year of age in mothers who had reported infection early in pregnancy, but had increasingly higher levels of choline at 16 weeks gestation. And again, that's just starting into the second trimester, so very early in pregnancy. And then finally, the data that I wanted to discuss with you, because marijuana was legalized for both medical and recreational use many years ago now. I think in medical use was legalized back in 2008 or 7, and recreational use around 2015. We've definitely seen an uptick in at least reporting of marijuana use over the course of pregnancy in our study participants and kind of generally in prenatal care practice. So we looked at mothers who opted to continue marijuana use in spite of recommendations at or beyond 10 weeks gestation, compared to women who reported no marijuana use at any time point during pregnancy, compared to those who stopped once they realized they were pregnant. So we kind of lumped them into three groups. The red line is women who use marijuana at 10 weeks or more. The blue line is no marijuana use over the course of pregnancy. And then the green line is women who realized they were pregnant, and this commonly happens in my clinical practice. Oh, I didn't realize I was pregnant. Okay, I'm going to stop using alcohol, using marijuana, smoking, etc. And again, looked at those choline levels at 16 weeks gestation. And I didn't mention before, but I'll mention it now. We also looked at choline levels at 28 weeks gestation in both that infection cohort that I just told you about and in this group. But by then, and that's just in the beginning of the third trimester, it does not appear to matter as much as earlier. We found that as maternal choline levels in their serum increased at 16 weeks gestation, the more buffering of P50, or the more good and adequate P50 inhibition we saw. So this is the group who used no marijuana, and their P50 scores are around 0.5, so normal in general. The no marijuana use may have had a slight influence, although we didn't have the numbers for this to be significant. But as their choline levels increased at 16 weeks gestation, their children were equivalent to the no marijuana users. And even the women who continued to use marijuana beyond 10 weeks had kind of a positive buffering effect on better P50 inhibition at 1 month of age in their newborns. And this also played out into the infant behavioral questionnaire, which this one was actually done at 3 months of gestation. I have 1 month, but it was at 3 months of gestation where we started to also see a similar pattern where higher choline levels at 16 weeks of gestation translated into improved IBQ scores at 3 months of age, even if women continued to use marijuana beyond 10 weeks gestation. So an unexpected or unanticipated finding, although one we are hopeful about as we continue to do a pretty concerning grand experiment in Colorado and other states with marijuana exposure during pregnancy. So this is an older slide, and I mainly put it in just to give you an idea of how TRNA7 and the cannabidiol-1 or CB1 interneurons are co-expressed. So on the left side is expression of the the alpha-7 receptor or TRNA7, and this on the right side in white is the gene that produces the CB1 receptor, and you can see that they are almost identically co-located. So CB1 or cannabidiol-1 is one of the receptors that is stimulated particularly by THC, tetrahydrocannabinol, which is the active or psychoactive component of marijuana. And so some kind of compelling histologic evidence on how marijuana may adversely influence TRNA7, alpha-7, informed or related neurodevelopment. And so luckily, we have been fortunate for a couple of different journals and press articles to pick up on our research. So this was an article back in 2017 in Science Magazine where they highlighted things that can play a role in reducing the risk of psychosis later that happen prenatally. So genetics, as I mentioned, serious second trimester infection, being born prematurely, which is a little bit more difficult to control, as well as birth complications, but then buffers, overall good prenatal care, and choline prenatally. So just to kind of summarize it, prenatal choline we know improves cognitive function into adulthood and decreases the risk of impaired neural programming and development in relation to the conditions that I've talked about: maternal marijuana use, perinatal infection, or a genetic predisposition to schizophrenia, but in other groups, data to also support a buffering capacity of choline in ADHD development in individuals affected with Down syndrome, which is trisomy 21 or 3 chromosome 21s, and in fetal alcohol exposure. So these are some different formulations available on the market for choline supplementation. Again, we aim for about 900 milligrams of choline, and we use this form, phosphatidylcholine, or Phoscol, in our studies. One, because we have to have an FDA IND to do this study, and two, because phosphatidylcholine is likely better absorbed. If you have a mom with some other condition that could be influenced by her microbiome or her absorption or her stress, than choline in its native form. And so for research purposes, that's what we selected to use. The downside is it's about $1,000 per pregnancy to take this supplement at the dosing we're using. These other brands that exist online or in your local natural food store sometimes would cost about $260 to $300 per pregnancy. And then the soy lecithin granules is available as a health food supplement, and it would be about $260 per pregnancy. It's a vegetarian, reliable vegetarian form of choline, and should be mixed with food, such as soup. And then if you're just interested in increasing your choline prenatally or non-prenatally in your diet, these are foods that are rich in choline, and this is quantified as milligrams per 100 grams, with smoked salmon being one of the highest choline-containing foods, as well as fried eggs and hard-boiled eggs, beef liver and chicken liver, which I laugh that about 2 pregnant women a year say, oh, that sounds great, I love liver, and everybody else kind of turns green when I mention that as a possible way to supplement. But there it is. And then there are some vegetarian sources and vegan sources, but you need to eat a lot more Brussels sprouts or cauliflower to get to that 900 milligrams than you would with a couple of eggs a day. So the foods that I mainly recommend for pregnant women are eggs, are salmon, because that also contains a lot of great omega-3 fatty acids, and salmon is a low to no mercury fish, so safe in pregnancy, and then chicken or liver if they can tolerate it. So to kind of give you a summary of how we're tackling this from the prenatal side, in standard prenatal care, we incorporate assessment of genetics and chromosome conditions and carrier screening for all sorts of conditions that can occur in an autosomal recessive or autosomal dominant form. We already recommend folate supplementation as part of our pregnancy nutrition, as well as prenatal vitamins that contain iron. And hopefully choline will become the big sister of folate someday. We recommend modification of the environment, smoking cessation, alcohol cessation, improving lifestyle factors like physical activity and sleep, addressing any infections or immunologic factors that we're able to, as well as addressing and treating psychosocial factors that can adversely influence the mom and the baby. And back in 2017, the AMA, or American Medical Association, even took a stance on choline that recommended that choline supplementation be included in an evidence-based amount to prenatal vitamins. Now, thinking through prenatal vitamins and what women are able to tolerate, especially in the first trimester of pregnancy when nausea and vomiting is highest, our group kind of believes that choline supplementation would be its own supplement or its own dietary changes plus supplementation, because most prenatal vitamins today contain 50 to 80 milligrams of choline, and what we're recommending is closer to 900 milligrams. These data and this stance was mainly taken in relation to the data on alcohol, fetal alcohol exposure, and how choline appears to have a buffering effect related to that. And I didn't present those data today. And then kind of the prequel, or what I'd like for you to consider as we're trying to expand this notion of choline being something efficacious and safe to take during pregnancy. So this kind of starts back with the story of folic acid. And various studies over time, or really over about 20 years, first looked at folic acid deficient diets, then adding dietary fortification, and then the inclusion of both dietary fortification and supplements to reduce the risk of open neural tube defects with folic acid. And an open neural tube defect, or spina bifida, is something that we can diagnose prenatally, and that you know how it's going to impact the fetus or newborn pretty soon after birth. And because the risk of these kind of birth defects were so markedly reduced with the introduction of folic acid supplementation, they were pretty widely adopted. And this is a figure that shows from the CDC the changes in neural tube defect prevalence before and after folic acid fortification. In the U.S., we went from about 10 per 10,000, or 1 in 1,000 births affected by open neural tube defects, or pregnancies affected by open neural tube defects, to about 40% less than that, or 7 per 10,000. So almost an order of magnitude as much. But then in places like Chile, they've reduced neural tube defect prevalence by 50%. And I think one of the reasons why is because this can be a devastating birth defect, and you have kind of the instant follow-up. You can see it on ultrasound. You can see it at birth. You see how it afflicts the individual immediately. And when we're dealing with manifestation of mental illnesses, there's a delay there. And so obstetricians have a harder time kind of focusing on the long-term benefits of choline versus folic acid. But if these types of changes or risk reductions could occur with schizophrenia or bipolar disorder or ADHD or autism spectrum disorder because of choline, then the population impact could be quite large. So I would like to acknowledge my mentorship, our research crew, the various labs that we worked with to get these data, and then wanted to give you a few takeaway points. One, that choline buffers the fetal brain. It looks like it buffers it from adverse genetics and environmental exposures that can predispose to mental illnesses and or behavioral issues that can impact an individual's and a family's overall life course. It could be part of primary prevention for the child, and this could translate over time into improved health for our society with much more thriving and much less struggle. So thank you so much for tuning in, and I'm happy to try and answer some questions.
Well, Camille, thank you for the work that you're doing and for really an outstanding presentation on a complicated but very, very important topic. I'm curious, when you speak to pregnant women about this, how do they respond? What's the typical response people have to the recommendation to take the choline supplementation?
It's typically very positive, and so I do in clinical practice extrapolate it to some other issues that we are still researching, because luckily, for research purposes, I don't frequently overlap with one of our research participants in clinical practice. But in general, when I say, hey, we have data that supports that this supplement is safe for you and your baby, that there may be benefits, or in particular, if I have a woman who tells me, oh my gosh, I was using marijuana or drinking alcohol before I knew I was pregnant, is there anything I can do? Then those are the perfect audiences and motivated audiences to go with choline supplementation. So then I give them the same dietary advice that I just presented and talk to them about the available supplements as well, what we're targeting in our research studies. And even in our research studies, we give everybody this information. We make them well aware that they will be randomized to receive the phosphatidylcholine or placebo, but also give them a little booklet that is very similar to that little dietary slide that I showed. And people are, I think, universally positive about it. I haven't had anyone say, no way, I'm not going to do that, or that sounds like a horrible idea for prenatal use.
Do you have any information on how widely recommended this is, in practices around the United States?
I know that it is not that widely recommended. We try to get the word out through various channels, and I think locally in the Denver metro area, or anyone that I've interacted with in Colorado may be more aware of it, because I'm a little bit of a I preach it a little bit, especially in vulnerable populations. But even though the AMA put that statement out in 2017, I think the uptake is slow, although not that much different than the uptake of other kind of interventions that we encounter in prenatal care.
Some of the work will take years to prove for sure. I'd like to speak a little bit about those studies, even some of the initial studies that the children are now a little bit older, but there's still, more time to go until they're at the age that schizophrenia might typically develop.
Right. Yes. Thank you so much for that question and that point. So the pilot randomized trial, which was 100 women, and those children were followed out to 40 months, took almost 10 years to complete. And that's been, those 40-month-old data were published in 2016. So this is something that Randy Ross started back in kind of the early 2000s. It's been a little bit faster recruitment this time since we have more support for choline, but it's still going to be a study that takes 8 years to complete. So at least 5 to 6 more years from now that we have reached that primary endpoint. And so we're kind of hopeful with the observational data that we have and the other findings, one, that awareness about choline and buffering these other kind of exposures like marijuana, potentially like alcohol, will be more rapidly adopted than might be by waiting on our additional randomized trial data. And two, we've obviously applied for different kind of funding sources to accelerate these investigations, not necessarily in a randomized fashion, because we don't feel like it's ethical to randomize women to marijuana use versus not in pregnancy or those kind of exposures, and would never pass IRB, and we'd never think that it was even reasonable to ask that. But to try and look in more pragmatic ways at women who continue to use marijuana or use alcohol in pregnancy and supplementing them with choline and still starting to follow their children out longitudinally like we have these other participants.
How about for people with above and beyond those risk factors of alcohol or marijuana use, a family history of schizophrenia, et cetera? What's the status of that?
Yeah, so those women, they can still participate in our studies, although a lot of times, like today, for example, I'm in one of our clinics where we're not recruiting, so I don't overlap with study participants at all. And I had a woman who has schizophrenia, and she is treated, but I just went ahead and made recommendations for choline supplementation in her because I feel like our data are strong enough, and there's no detriment that we've identified that it's warranted in her and for her child.
Now, you gave the extraordinary statistics for the effect of folic acid on the neurotube deficits and the significant decrease. Do you have any sense about what the effect may be for this intervention for schizophrenia, maybe some of the other psychiatric conditions?
Schizophrenia is maybe the hardest one to answer because none of the pilot randomized trial kiddos are approaching early adulthood yet.
Right.
So I think it would be more based on the infection data that we have and the marijuana exposure data. But those data suggest, and they're observational, they're not randomized, that it essentially eliminates the detrimental markers or the findings that we can get early, at least at one month of age, 3 months of age, 1 year of age, 4 years of age. And so it will be interesting to see how that plays out. And I'm optimistic that if we aren't seeing adverse effects early in childhood, that hopefully those children have kind of escaped that critical period of fetal brain development that is involved in whether this goes well or whether it goes poorly. But
right.
I can't say kind of the I can't give an order of magnitude or a risk reduction number. Although right now it looks like close to complete mitigation.
So, and this is based on the auditory EEG, and
this is based on the auditory EEG, and then either the infant behavioral questionnaire or the child behavioral checklist. Similar assessments for different ages.
So that all of the people that were in the active receipt of the choline supplementation didn't show any of what would be viewed as the negative potential signs on those types of evaluations.
I think essentially all. There may have been one or two still, and there were definitely some who received the supplement or didn't. There were always a handful in either group, but in general the trend was that choline improved the P50, the auditory gating study, as evidence of better inhibition or completion of that last step of GABA becoming inhibitory during fetal brain development.
So I'm just going to summarize, because to me there's so many important take-home messages from what you just presented. First of all, the relationship between very basic science, the genetic work, and looking at some genetic studies and what those genetic studies may mean in a developing brain, and then what an intervention can be, which really shows the thinking of science and discovery, and bringing that to actual real-life interventions that potentially could have a tremendous impact on individual lives and on society in terms of these very important, significant illnesses. And the work goes on, but at this point, for a pregnant woman, your recommendation is that they take the supplementation.
Yes.
Well, I can't tell you how appreciative I am of your work and the work of the team, and how important it is to get the word out about this. And I want to thank you for all that you've done and all that you continue to do on this extraordinarily exciting research that you've been involved with, and that you've really helped to lead. So, Camille, thank you very, very much.
Thank you so much for allowing me to speak today.
Yeah, well, thank you. I also want to thank everybody who joined us today. All of the research we fund is made possible through private donations. So please consider making a gift by visiting bbrfoundation.org or call us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with family and friends, please visit the events and webinar page on our website. I hope you'll join us again next month when Dr. Helen Mayberg, senior faculty in neurosurgery, neurology, neuroscience, and psychiatry at the Icahn School of Medicine at Mount Sinai, will present Deep Brain Stimulation for Treatment-Resistant Depression, a Progress Report. This webinar will take place on Tuesday, October 15th at 2 p.m. Eastern Time. Thank you again for joining us. We appreciate your support. Take care.